Assalamualaikum and good day everyone. So for this week we're moving on to a new topic. Again, we are almost towards the end. So this is on post-transcriptional regulation. This would be more or less the last control in regulation of, or basically to regulate your gene during the transcription. So there are 6 mechanisms, or basically 6 things, 6 factors, parameters that you could control. First is basically we start off with RNA splicing. Yes, but what I have for you here is actually alternative RNA splicing, because we know RNA splicing occurs, but again there is an alternative to it. What are those? Okay, let's have a look. So we know that, okay, this is a bit of the history of RNA splicing. So it was first discovered in 1970s in viruses, and of course scientists later on discovered alternative patterns of pre-mRNA splicing, which produce different mature mRNAs containing various combinations of exons from a single precursor mRNA. So the first example of alternative splicing in eukarya is actually the IgM gene, which is a member of the immunoglobulin superfamily. So alternative splicing, or sometimes you see the acronym as AS, is actually a process whereby exons or portions of exons or your non-coding regions within a pre-mRNA transcript are basically differentially joined or skipped. So this will result in multiple protein isoforms, which are encoded by a single gene. So it would increase the information diversity and functional, basically make sure or giving your gene to have a diverse function sometimes, or more or less an alternate function to its original function. Now, more or less, this is how alternative splicing could look like. So this is a general overview. So when you have your normal transcription, so usually you have your gene meeting in, so it is being transcribed. So you know that you have different exons over here, and your introns will be cut off. That is in the actual RNA splicing. But in alternative splicing, for example, over here you have 5 exons. So if alternative splicing occurs, you may have mature mRNA, which have only 4 exons, or basically, and they are in 2 forms, which we refer to as isoform. So isoform A, whereby exon 3 is being skipped, and it is being translated to produce a protein with a function A, because it doesn't have exon 3. Probably exon 3 would have or would help with a certain function, so no, it doesn't have the exon 3, so it have another function. The other isoform is whereby the next exon is being skipped, which is exon 4. So when being translated, so you have protein isoform 2 after translation, which has another function, probably a bit different function as in the first protein isoform. So this is what alternative splicing do, and there are different variations that it could occur. So what is the basics? So basics, of course, starts with splicing process, and we know this is a splicing process, so I'm not going to repeat again. We have covered this in quite detail previously. So apart from that, we know that splicing can also occur by other methods such as RNA strand forming ribosome, which by the RNA actually act as the enzyme to cut the strand. This is called as self-splicing. So additionally, specific cells also have self-editing mechanism, whereby they could determine their own specific protein shapes and basically how the protein isoforms could be, and this is mostly true in plant and fungi. So these are the different types of alternative splicing. So this is the most common that we know. So by right, all your exons, after the introns have been cut off, spliced, so they are joined together, you have all the exons in a typical mRNA transcription. Alternative splicing, this is type number 1, involve an exon, which is usually considered a piece that will stay, being spliced out, or what we refer to as exon skipping. So another one would involve 2 exons in a series with only one of the 2 being kept in an alternating pattern. This is what I've shown you early on, in the general overview, whereby number 3 and number 4 being skipped. So you can get different isoteins being translated later on. So there are also a number of types of alternative splicing patterns where different parts of the introns are cut. Sometimes mutations near the edge of the intron can affect this time of process. So that means that it would sometimes signal for it to be cut off or to be included in the splicing process, or during the ligation to produce your mature mRNA. And another type is where the intron is being retained. So there are occurrences where the introns are being retained. So what are the mechanisms of alternative splicing? So when it occurs, cis-acting regulatory elements in the mRNA sequence would determine which exons are retained and which exons are spliced out. So these elements would alter splicing. This is done by binding different transacting protein factors such as SR or serine arginine-rich proteins. And serine arginine proteins actually has a function as a splice facilitators and heterogeneous nuclear ribonuclear protein, which suppresses splicing. So when inhibition of silencing occurs, so it could actually be achieved sterically, meaning that when it binds when it binds to the splicing inhibitors, so the splicing silencers that is located in close proximity to the splicing enhancers could block the binding of SNRPs and other activator proteins or prevent spliceosome assembly. And this would in turn more or less inhibit the splicing. So the final decision to include or splice an alternative exon is actually determined by combinatorial effects, which include cellular abundance and also competitive binding between SR and also HNRNP inhibitors. So both activators and inhibitors. And the outcome of alternative splicing depends on the stoichiometry and interaction of splicing activators and inhibitors, as well as we know steric conformation and accessibility of the splicing sites. If there is mutation, then of course it would play havoc there. So the splicing code basically is important or basically have more or less an impact on the alternative splicing, and research has shown that there are hundreds of characteristics of RNA structure, and this include regulatory particles as well. So protein or microRNA can determine how this alternative splicing occurs. So before all this was basically studied, it was thought that the codes at the edge of the introns are the most relevant because it would signal for splicing. But it appears that the structure deep in the intron itself, or from the cut, may be quite relevant as well. So there are dramatic differences in splicing which are triggered during the different phases of embryonic development. So these differences occur among different types of tissue cells and not to mention organisms. So apart from that, there are also other factors that affect splicing pattern, which include transcription rates, co-splicing machinery levels, competition between splice sites, chromatin structure affecting the rate of transcription, histone modification, and local chromatin binding. So how is alternative splicing important? So even though it is suggested, at least 75% of around 30,000 human genes that have been recognized undergo alternative splicing, and this result in 2 or more splice isoforms with striking variation across tissue types and developmental stages. This would give us whereby we are different from one to another, even siblings. So recent advances in high throughput technologies have of course facilitated studies of genome-wide alternative splicing, whereby more than 95% of human multi-exon genes would express multiple splice isoforms, and the evidence are often differentially regulated across tissues and during development. So this is also true among individuals and population, which suggests that individual isoforms may serve specific spatial and temporal roles. So alternative splicing is of course known to be involved in the regulation of normal physiological function, and it is also known to be or to have an impact on pathologies. As an example, a number of alternatively spliced genes in immunity are known. So, for example, you can actually read on about it. So, the studies have indicated that alternative splicing of CD44, a gene involved in T cell homing with 10 variable cassettes exons and 6 distinct protein isoforms, is crucial for T cell function. And we know how important T cells are. So, current technological advances suggest that alternative splicing is more widespread than initially thought, and this would be likely to be involved in a number of human pathologies and probably other organisms as well. So I actually wanted to make 6 videos, but I think that would be a bit lengthy, so I'll basically just move on to the next factor, which is on polyadenylation. So how polyadenylation could basically add another level of regulation. So there are 2 steps process in polyadenylation. So it involves specific endonucleotide cleavage at the site determined by the binding of the polyadenylation factor, and it also involves polymerization of the adenosine tail to about 200 residues in higher eukaryotes and around 70, okay, basically amino acids, okay, as in yeast, okay. So the tail length is of course organism specific. So mostly it's quite longer, okay, in plant and also animals. So almost all eukaryotic polynylation signals contain the core upstream element, okay, which is the consensus sequence AAU, AAA, okay, or sometimes a variant. So this is about 10 to 35 nucleotide upstream of the actual site of poly A addition. So the sequences of about 14 to 70 nucleotides downstream, okay, which is known as the core downstream element, are known to be involved in directing polyannulation. Apart from that, there are also auxiliary elements upstream and downstream of the basically AAU AAA sequence, which have been characterized, and it could enhance polyannulation efficiency. And this, of course, again, is often mediated by specific binding of additional transacting factors. So most basal mammalian pollination and cleavage machinery comprise of 4 basal subunits protein factors, which are assembled on the RNA before any reaction takes place. So in a tightly two-couple step process, cleavage occurs at around 10 to 13 residues after the co-upstream element, and this is followed by addition of poly-A tail by poly-A polymerase. Now, how is polyannulation used to regulate gene expression? So basically it depends, okay, and it is basically used to regulate in a variety of developmental and proliferative way. So most often, several auxiliary factors bind to the pre-mRNA, and they either compete or cooperate, or sometimes both, okay, with each other to modulate the mechanisms of cleavage and polyannulation. So there is also small alterations on the concentrations of any of these factors in the particular physiological state of the cell, and this would affect the outcome of the pre-mRNA 3' end processing, and well would have certain impact, okay, on gene expression. So now, so it's actually covering up, okay, this one, but never mind, okay. So basically, okay, as you can read on about it and you can also look at it, okay. So many, okay, RNA cis-acting elements usually are present at the 3', okay, UTR, and this can affect pollination, okay, or other RNA processing or RNA mediated event. So regulation of gene expression at the 3' UTR depend on the presence of cis-acting regulatory element and may influence 4 of these things, okay, or sometimes all of them, or in combination. Nuclear and cytoplasmic pollination, which is pollination signal and cytoplasmic pollination element, CPE. Stability of the mRNA produced, which we will cover again in later part of this topic. The subcellular localization of the mRNA, and of course translation or mRNA decay. So the presence of microRNA target sites in the 3' UTR, which may influence gene expression, may not, okay. So the presence of trans-acting factors, okay, on the other hand, will bind to these elements, or sometimes the microRNA that target the mRNA in the cells or tissue. So this is basically occur at particular stage, and at the correct concentration, it is of course essential for regulation to occur. So basically, alternative pollination, just like your alternative splicing, okay, which produce isoforms, okay, containing varieties of your mRNA with the tail, with different tails. So for example, over here, alternative polynylation of mRNA, as shown in the center of the figure, okay, can affect many other cellular or organismal events, such as some of these. Now, how do we classify alternative pollination? Okay, so there are different types. Okay, we'll start with type 1. So only one pollination signal is present at the 3' UTR, resulting in only one mRNA isoform. So this is more or less, I would say, quite common. So type 2, okay, whereby, okay, more than one polyannulation signal is present, but, okay, there is a but here, only one is present in a common terminal exon. So what it will do, okay, more than one resulting mRNA would be produced, but with no effect on the encoded protein. However, sometimes due to possible alteration of mRNA stability or transstability, other downstream effects, okay, downstream effects, so there may also be changes to the amount of protein produced. So if this occurs and if it is chosen. Type 3. So type 3 is divided into 2. Okay, so altogether you actually have 4, okay, including type 1, type 2, and type 3. So type 3 is an alternative pollination signals that are present at the upstream introns or exons. This would invoke alternative splicing along with alternative pollination. Okay, so the first type, okay, is actually type 3I, okay, which is intronic alternative pollination signals, and type 3E. So I and E, E is exonic alternative pollination signal. So both of them may or may not result in different protein products being produced. So it would depend on the stability of the mRNA and the presence of an in-frame stop codon and the overall translation competence of the mRNA. So these are the four types being shown in one diagram, and you can basically just see that. Okay, so this is the first type, whereby this is the most common. Even though this is type 1, or being considered as alternative splicing, this is the most common that occurs if there is no other alternative being provided. So you have your typical only one site, for the stop, and whereby signals for pollination, to add your poly A tail. Type 2, is whereby you have more than one signals. So what will happen, is that, well, basically it has more than one pollination signals, so it can either choose this one or this one. So that means that it may add your signals somewhere here or somewhere there. So it depends on which one is being chosen. So for type 3I, which is intronic, so you can see that you may have a premature stop signals, and basically to add the polyannulation or poly A tail. So for type 3E, which is on the exon, basically sometimes the poly A tail signals is before the stop. So this could sometimes result in isoform. So these are some of the examples, but there are consequences to this. So I would say more or less for this two, it wouldn't really have that much effect on your mRNA or basically on your gene, but these two, to a certain extent, do have a certain effect on the gene and also translated protein later on. Now, basically how alternative splicing and coordination with other RNA processing events occur. So we know that all processing RNA events are intertwined and interconnected. So these include interactions between pollination and splicing, transcription termination, translation, export, and also stability. So protein and RNA factors involved in many of these interconnected processes. So they are now known to be involved in regulating alternative splicing and alternative pollination. So for example, the NOVA, a well-characterized splicing factor which regulate APA in brain. So you can actually read down about it. So that would basically signify the end of the alternative pollination. So I'll stop here. So move on to the next video, to see what are the other remaining 4 factors involved, to control regulation in post-transcriptional control.